started. Hi, everyone. Sorry for the dogs. Hello, welcome. Come on in. My dogs can be very loud at times, and hopefully they'll be well behaved for us. Come on in, everybody. We'll give everybody just a couple minutes to join us.
I still haven't fixed my computer. I still say owner. It's me, Vicky.
Oh.
And I also opened up those 2 quizzes, which I couldn't do because I hadn't had the lecture yet.
Oh, okay.
I'm so stupid.
That's all right. It takes a little while to get used to how everything works. I know. So.
I am not sure if the quizzes or if they're announcements. So I opened it and I was like, oh, crap.
I'll adjust it so that that's all right, so that you can
Okay.
get back in.
All right. Thank you.
Not a problem. I know it takes a little bit to get accustomed to all the information that gets thrown at you at the beginning of the course. Let's see if I can adjust this a little bit. Well, I have my laptop back, which I'm thrilled about. I felt like I was walking around without one of my arms for a few days because I didn't have my laptop. So
it's awful, huh?
It's terrible. You don't realize how dependent you are on them until you don't have them. And I was lucky because I had access to a tablet, but it just wasn't right. It wasn't my laptop. It wasn't right. So I'm very happy to have it back. And I'm happy that it wasn't too expensive a repair either. Because as I'm sure you know, it can get very expensive very fast. So I lucked out there. I'm just going to adjust this. All right. I'm just taking stock of who's here. We'll give folks another couple minutes. We're just still right before 4 o'clock. So this is really our first full week of class. And if you remember, I said when we were together last time that things are a little more busy this week than usual. Usually you have 2 lecture topics, 2 lecture quizzes, and then a lab homework assignment. And this week, because 2 of the lecture topics are short, they're put together and you end up with 3 quizzes, 1 for history, 1 for microscopy, and then 1 for the cell. So it's a busy week, but then we'll settle right into our regular weekly schedule after that. So, all right, go ahead if you would and just type your first name into the chat for me so I have a record of who's here today. We'll do that at the start of all of our lab meetings, just so I have a way to look back and see. I was happy to see that everybody got their work in for the first week, which is great. I think there was only one student who was missing one thing. If you remember, you had to hand in your student survey and your first lecture quiz and your first lab questions for me by Sunday at midnight. And just about everybody got everything in, which is great. And I was also really pleased to see that most everybody did very well on those first couple of assignments, which is also great. And you'll get more accustomed to how I ask questions. And as we go, I try to ask the style of question, the type of wording that I would normally use, like on an exam question, so you can get used to that. The other thing you should know, if you haven't noticed already, is whenever you go back into an assignment in Canvas after it's been graded, you can click on a little link, I guess, that says View Instructor Comments. Because as an instructor, I can type in a comment directly under a question on your quiz. And I do that quite frequently, especially if you've missed something and I want to point out what you might have missed, or maybe you got something right and I just want to tell you, or I want to add a little something into your answer. So it's always worth your time to go back after things have been graded and click on that View Instructor Comments, wherever that it shows up on your view, and see what else I've written for you. Like I said, just about everybody did quite well on the quiz and the lab homework questions. There were a couple questions that I made note of just to mention. For example, one of the questions about using the microscope was something about what are the parts of the microscope that that determine how much light gets to the eyepieces. And the correct answer for that question was actually 3 different things, because there's a light source on a microscope. Down at the bottom, at the base, there's a source of light. Then there's the iris diaphragm, and then there's the condenser. And all 3 of those things are underneath that stage, that black stage. Now, if you told me any one of those, I gave you credit for the question. Just starting out, that's fine. But just so you know now, there are actually those 3 controls that we can adjust and play with whenever we look through a microscope. So we can really fine tune the lighting that comes in over an object on a slide. It's not easy to know to use them correctly. More often than not, we're stuck trying to figure out what's wrong with the condenser, like why it isn't set correctly. But with practice, both the iris diaphragm and the condenser are super helpful for really honing in the light just the way you want it to help you see what's on your slide under the scope. That was one question I want to mention. The other one, there was in your lab homework, there was a question that was labeled clinical application. It was a 2-part question. I try to put some of these clinically based questions on your homework. These are thinking kind of questions, and they can be a little tough. I thought this one was a little tough. If you remember, it was a question about a patient who had a pneumonia, and a sample was recovered from the patient and examined under a microscope. And you were told that you could see microbes at 40x, and you could see them even better under the 100x lens. And the first part of the question said, why do you think, I guess I also told you in the statement that you decide that it must be bacteria. So the first part of the question asks, how do you know it's not Archaea? About half of you got it right, which is great. The answer to that question is you can be very confident that it's not an Archaea that is causing this patient's pneumonia, because there just aren't any Archaea that we know of that cause disease in humans. They are amazing organisms. We're still learning a lot about the Archaea. Several of you told me that Archaea are extreme organisms. They tend to live in extreme environments. That's very true. I did want to point out that it's not that humans and Archaea never come in contact with each other. In fact, as we learn more about our microbiome, our collection of microbes that live on us and inside of us, we're starting to learn that there are some archaea that are part of our microbiome. But again, there isn't a single species of archaea that is known, at least right now, that causes disease in humans. So that's how you know that that could not have been an archaea that was causing that patient's pneumonia. It had nothing to do with how you would view that organism. Some of you tried to tell me that archaea look very different at 40x or 100x than bacteria do. That's actually not true. They look very similar. They're a similar size, they have similar shapes. Structurally speaking, unless you were looking through an electron microscope, it would be very difficult to tell bacteria and archaea apart. So for that question, it was all about just a logical deduction. It couldn't be archaea because this is a sick patient. It would have had to have been a bacteria that was making her sick. Now, the second part of the question asks, how can you be sure, that it's not a virus? A lot of you got this correct, which is great. You told me that it can't be a virus because if it were a virus, we wouldn't be able to see it under the light microscope. That's exactly right. We would not see it at 40x under the 40x lens. We would not see it under the 100x lens. Can't see a virus with a light microscope. You have to go to an electron microscope. Remember, viruses are in the nanometer size range. That's 1,000 times smaller than a bacterial cell. So very, very small particles viruses are. So that's a helpful thing to remember, that anytime you're looking under a microscope, if you see little pinpoint things that you think are microbes, at least you know they're not viruses. If you're seeing them with a light microscope, it's not a virus, too small. So Debbie's asking a great question. Debbie's asking, would it be possible to find archaea in the lungs of a human, even if the archaea weren't causing disease? The answer, Debbie, is we don't think so, but we honestly don't know yet. We don't know the extent of archaea in our microbiome. Now, we don't expect to find any microbes deep down in the lungs. So let me say that first. You would not expect to find any microbes down in the alveoli. And that's because we expect the alveoli, or the air sacs in the lungs, to be sterile. But there are some microbes that can be found in the tubular pathways in the lungs. So the bronchi and the bronchioles can sometimes have microbes present on the epithelial surfaces. I doubt personally, I just personally doubt that there are archaea living there from what I know of the archaea. But I'll bet you that we're going to find more and more archaea in our guts. I think there are probably a fair number of archaea that normally inhabit our gut, our intestines, in other words, and are part of our healthy microbiome. But we just haven't done the work yet. The research just hasn't been done yet. We just don't know yet the extent of the archaea that are on and in our bodies, which is kind of interesting. Good question. Yes, that's right, Debbie. We do find archaea in some very extreme places. And you don't think of the human body as being extreme. We do find some species of archaea in extremely hot places. We find some species in extremely acidic places. We find some species in very salty locations. These are places that bacteria just couldn't survive. But the archaea have evolved to take advantage of these little niches in the environment. But we may find that there are archaea that have evolved to find our bodies to be a nice environment too. We're really just in the infancy of knowing all of the different organisms that live, for example, in our intestines. We're just in the infancy of that. We're learning now which organisms belong in us, either not harming us, not really doing anything, bad or good, or actually helping us, actually being helpful to our health down in our gut. Good question. Yes, the microbiome is one of the most interesting areas of microbiology research that's sort of on the horizon right now. There's a lot of money going into that area right now because we really are beginning to have a more solid understanding that our health depends upon having a very wide range of microbial species living inside of us. And as I said, on us, on our skin, in our mouths, just all over us. Um, there's even some evidence that there are viruses that are part of our microbiome and should be there. They're part of the natural population of microbes that live on us and keep us healthy. All right, very good. So today, we're going to have a laboratory exercise about examining living microbes in the laboratory. And on Wednesday this week, we're going to talk about a technique that's just referred to as simple staining. We're going to begin talking about the techniques that we use to examine microbes in the laboratory. Now, when we need to examine a living microbe, you might ask yourself, well, aren't we always sort of looking at living things in a microbiology lab? And the answer is no. The answer is many times, in fact, most of the time, when we're doing diagnostic work in the laboratory, when we're taking a sample from a patient, for example, and we're trying to figure out what this microbe is that's causing them some kind of disease, most of the time, we're actually going to stain that sample. We're going to prepare a microscope slide and we're going to stain that microbe. We're going to apply any of a whole variety of different colors of stains to help us visualize that organism more. But there are times when we don't want to stain. And that is because the stains that we use actually kill microbial cells. And if you can imagine, there are applications in human medicine and in veterinary medicine when we suspect that a patient might have a microbial infection with an organism that is mobile, with an organism that's capable of swimming, if you will. And there are several well-known pathogenic microbes, bacteria particularly, that are capable of movement. They have those long extensions coming off of their cells called flagella. So if we were to kill those by staining them before we examine them, we wouldn't be able to see that mobility. And if we view them while they're alive, we can. So there is a time and a certain circumstance in the laboratory where it makes sense for us to examine organisms when they're alive, when they're living. Sorry, I dropped my stack of papers here. So that's what we're going to be talking about today. Before I jump into the slides, I just want to ask if anybody has any questions about anything at all. Any questions about our schedule this week or something you may have gotten wrong on a quiz or anything like that? Go ahead.
Hello.
Yes, hi.
Hi. I have a question about the future exams that we're gonna have.
Yeah.
So as far as notes, I obviously take them on all the slides and what you say and everything.
Yep.
As far as the pictures, how are those going to be on the exams? Can we print them out and tape them to our notes? How does that work?
That's a hard question. A lot of the times I use pictures, images to drive home a point.
Okay.
But sometimes there are images, I don't know if you've seen many so far, but sometimes there are images of particularly structures in cells that have names, and you need to know the name of a structure.
Yeah.
So I would say go ahead and either print out images and put them in your notes if you find that helpful, or make your own sketches in your notes if you find that helpful. I will generally let you know if there's something, particularly in a microscopic image, that I want you to be able to identify on an exam.
Okay.
Yeah, I generally will let you know if you need to be able to recognize something in an actual photographic image under a microscope.
Okay. Thank you.
Yeah, no problem. Good question. All right, so
I have
I'm
a
going to,
question too.
oh, sure.
So, I saw when we take the little like 5-question quizzes that it says that you have like endless attempts. Now, say you go and take the quiz and you get like 1 or 2 wrong, and then you go take it again. Which grade do you take?
So that's a great question. I'm glad you brought that up because it's a little confusing. So each week you're going to be doing lecture quizzes for me, and you're going to do a set of laboratory questions. Now, they're both referred to as quizzes in Canvas, but really only the lecture quizzes are true quizzes. And by that, I mean you're going to be timed. You have 30 minutes to do the 5 or 6 questions, and you have one go at it. So you sit down when you're ready, when you've watched the lecture and you have your notes compiled and you're ready to go. You sit down with your notes next to you, and you go into the quiz and you take the quiz. That's generally a 5-point assignment. Again, it's timed. You have 30 minutes. And you have one attempt at it. Now, I mentioned in the quiz last week that if you look at the settings, you'll see that it says multiple attempts are allowed. Now, that is set like that for reasons of people sometimes accidentally going into a quiz and when they're not ready to take it. It happens particularly at the start of the course. People sometimes will click on the quiz. They didn't really mean to. They're not ready to take it. And you see, if I set it for a single attempt, they won't be able to get back in again. So I leave the quizzes, at least at the start of the course, I leave them set for multiple attempts. But I don't want you to take the quizzes more than once unless you've notified me that you made a mistake and you need a second chance. The idea is you're going to take it once because it's a practice for you before you have to take an exam from me. And they're sort of low stress in that they're only worth 5 points. So even if you miss a question or two, it's not going to impact your grade that much. So those are the lecture quizzes. Now, the lab questions, even though they're called quizzes, they're not quizzes. They're set up as a quiz because that's what Canvas calls them. On the lab questions, there's no time limit. You can go in and out of that as many times as you want during the week. You can take multiple attempts at answering something. If you answer it one way and you don't really like it, you can go back in and change it until you do your final, submission by the due date. You can keep making changes. That's really a homework kind of assignment. And all of the lab questions will be like that. So for any assignment that's called lab questions, you can take all the time you want. You can go in and do as many attempts as you want. You just have to make sure that you submit what you want to submit before midnight on Sunday. And that'll be what I grade. Does that answer your question? Trying to think if there's anything else I can point out.
Yes, it does.
Okay, good.
Thank you.
Now, when you take an exam for me, those are going to be timed, and you're going to have one attempt at those. Again, unless something weird happens and we need to give you a time to go in a second time, sometimes that happens. But for most of you, you'll go in, you'll take the exam, you get that one attempt. I will give you plenty of time. Don't worry about that. The quizzes are actually a nice practice, if you will, before you have to take the exam. All right. Anything else? All right, good. So I'm going to pull up the slides for this lab. And again, these are up on Canvas for you, just the slides though. You're going to see that we're going to watch a little bit of video together today. The video is not on Canvas. So this whole meeting will be recorded. Hopefully no more problems with Zoom now that I'm back on my laptop. Our whole meeting will be recorded. So if you want to go back in and you want to watch some video again that we talk through today, you can go onto YouTube and just watch this video again. Okay, but the slides themselves with the words on them are up on Canvas for you too, if you find those helpful. So at the start of each of our labs, I'm going to point out what our objectives are for the lab, why we're bothering doing this lab. I'm going to stop periodically and I'm going to ask for questions. So feel free, if you want to stop me, feel free to stop me, but know that I will stop periodically and ask for questions too. So today's exercise really has two parts. In the first part, we're going to talk about the techniques we use to examine living microbes, particularly bacteria. We're going to talk a little bit about microscopy because we're going to talk about the mode of microscopy that's called phase contrast. Now you'll find out a little bit about the different modes of microscopy in the lecture material this week. But for now, know that one of the things that a light microscope is capable of is called phase contrast, and that is a particularly good tool for examining living microbes. We're going to talk about making what are called wet mount slides. We're going to talk about making what are called hanging drop slides. And we're going to talk about the difference between true motility in a microbe being literally able to swim, to move, and what's called Brownian movement, which is a phenomenon that is sometimes mistaken for motility when you're looking under a microscope. Now, in the second part of the exercise today, we're going to make a wet mount together. We're going to go through, or I'm going to go through the process of making a wet mount so you can see it step by step. So remember, most of the time when we're examining microbial cells in a laboratory, we're actually going to prepare a microscope slide and we're going to stain the cells. And the reason we do that is because the staining that we do helps us see things better. And especially when we're using a light microscope in what's called bright field or what's called dark field, those are two other modes that you can use. When we're using those modes, staining helps us see things better. It just does. But we have to remember that when we stain our microbial samples, we're going to kill the cells. Now, we kill the cells twice, essentially, when we make a traditional stained specimen, because we have to heat up the cells to make them stick to the slide. That's what's called heat fixing. And then when we put the stain on, the stains themselves are toxic to cells. So we're going to kill it. If it's a living cell and we're putting it on a slide and we're staining it, we're going to kill it. Now, I mean, in a way that's nice, because once a slide is prepared and stained, we don't have to be as careful about handling that slide because the microbes are dead. So we can take our gloves off and we can examine it under the microscope. But like I said, there are particular times when we don't want to stain our samples because we suspect that we might have a microbe that is mobile, that's capable of moving under its own power. And there are, in human medicine, there are some well-known pathogens, particularly of the gastrointestinal tract, that have these structures called flagella that allow them to move through their environment. So if we get a sample in the lab and a physician suspects one of these fecal pathogens that can move, it makes sense for us to first examine it without staining it and see if we see movement under the microscope. Now, there are a couple of other times when using examining cells as they're alive makes a lot of sense. Environmental samples, for example. Here in New Hampshire, you know, we have a lot of recreational waterways that people are always using. We have lots of lakes and we have rivers and streams, and New Hampshire does a really good job at the state level of monitoring recreational bodies of water for pathogenic microbes, the kind of microbes that can make people sick. So the state sends out technicians to gather water samples periodically through the warm weather months. And those samples are analyzed to make sure that if there is a particular pathogen in that body of water, that we prevent people from going in and swimming and so on for a little while. So those kind of samples, those are often examined without staining them. We take samples from water treatment plants periodically and we examine those. People who live on well water, again, very common in New Hampshire. Sometimes samples are taken from wells or from above ground tanks, water tanks and cisterns, and examined without staining. Even things like in commercial buildings, like office buildings, it's common to take samples from ventilation systems, from humidifier systems, because there are certain pathogens that like to grow in those systems. And we frequently examine those without any staining. So let's say a couple things about phase contrast now. Remember, you will read or you will watch, I should say, more about these modes that we can use on a light microscope. You'll hear more about that in the lecture material this week. But there is a way to use a light microscope that is called phase contrast. And it's particularly useful when you're examining living cells. Without a stain on the slide, it can be hard. It can be hard to see the cell because the cell doesn't have any color. So you're looking at a colorless cell on a colorless background, and it can be really hard to distinguish the structures in the cell. Now, the nice thing about phase contrast is that it works in a way to allow us to see the three-dimensional structures in living cells. So it is specifically useful for identifying three-dimensional features of a living cell. Remember, cells are three-dimensional. We often think of them as being two-dimensional because we tend to look at them as though they're flat on a slide. But in real life, cells are three-dimensional. And living cells, especially those that are moving around on a slide, are going to be changing position in 3 dimensions. And phase contrast lets us see those different dimensions, those different levels of the cell through its spherical structure or elongated structure. It's all about a property in physics called refraction. I'm not going to talk about refraction in this course. It's frankly a little bit above my understanding, all of the physics of it. But if you remember from your high school courses, if you ever took a physics class, when light passes through different materials, it bends in different ways. So when it passes through air, when it passes through water, when it passes through living tissue, the light is going to interact with that substance differently in each different substance. And phase contrast takes advantage of that. And the reason you can see three-dimensional details so well in phase contrast is because it shows those differences, the differences between the cell and its surroundings. It shows them as brightness. So if you look at the top of this slide, over on the left-hand side, we're looking at a group of cells under phase contrast. There's some more cells over on the right. These don't happen to be a combination of microbial cells. There are also some eukaryotic, some human cells in this sample, but that's okay because the idea is the same no matter what kind of cell you're looking at. Notice how some of these cells have a very bright white ring around them. Now, if we were looking under the microscope at these cells, what we would do is we would very gently just roll back and forth the fine focus knob, and it would show the detail of the three dimensions of these cells as brightness. And that would allow you to say, oh, this is a cell. I know it's a cell because it's got that big bright ring around it. And, you know, something else on this slide might just be a piece of debris on the slide. And I'll be able to tell that because it won't have that brightness when I alter the fine focus up and down. So the thing to remember about phase contrast is that it's very useful when we're looking at living cells because it helps us tell the difference between a three-dimensional cell and something in its environment. We can see differences based on different brightness amounts, and it allows us to see these cells even though they don't have any color. We haven't stained them any particular color. Now, on this slide, over on the left and the right, you're looking at a couple of samples that were taken out of bodies of water. Now, these are kind of perfect samples in that we took samples and then we put them in a centrifuge and we gathered all of the material in the water down at the bottom of the tube so that when we looked under the microscope, like we're doing right now, you can see all of whatever was in that sample all in one place. So there's an awful lot of material over here on the left and over here on the right. And you can see some of this material has color, doesn't it? Some of this material is green. Now, why do you think it's green? Anybody have any ideas? What makes something microbial green? I didn't put any stain on it. Anybody have any ideas? What do you think might make
Is it plant matter?
Yeah, and what's in plants that makes them green?
Photosynthetic? Are they photosynthetic?
They are photosynthetic. Anybody know the name of that chemical?
Chlorophyll.
Chlorophyll, good. It's a pigment. It's a pigment. That's why it looks green. And there are photosynthetic microbes. There are little bacteria and there are other microbes like algae and protists that are capable of doing photosynthesis. And so they have chlorophyll in them as well. There are actually other pigments that you find in some microbes that are red colored, and there are some that are brown colored that also allow those microbes to do photosynthesis. So it's very interesting. It's very cool when you look under the scope at these particularly these water samples, you'll see some color, which is nice. You can see some brown, you can see the green, you can even see some sort of a gold color, some red. Those are just natural pigments. We didn't put anything on there. These, again, both came out of natural water samples. So it makes a lot of sense that there's a whole collection of microbes on here. Again, some of the very smallest are probably bacteria. There might be Archaea in here. There might be algae in here. There might be protists in here. There might be fungi in here. But let's say I was looking at a body of water, and I was looking very particularly for a pathogenic bacterium. For example, there's a type of bacteria that we find quite a bit in New Hampshire in our lakes and rivers called Cyanobacteria. We used to call that organism blue-green algae because we used to think it was an algae. But it turns out it's not. It turns out it's one of these very primitive bacterial species. And it can cause disease if it grows to huge levels in lakes and streams. And you've probably seen this sometimes when you drive around in the summer on super hot days. Sometimes you'll see a greenish slick on the edge of a pond. Sometimes that's algae, but sometimes that's cyanobacteria. And that's one of the things we monitor for. If we find that in a recreational water body, we're going to close that place down for a few days until that bloom of bacteria dies off and goes away. Now I want to show you this video. In the first part of the video, it's very hard to see anything. You'll notice that this has a bluish color to it. We're looking under the microscope here. Now we've gone up to a higher magnification, and now I think you can see something. The blue, by the way, here is not stain. What this blue is just a filter that's been applied to the eyepiece of the microscope just to help us see better. This is a particular type of bacteria called Enterobacter Erogenese. You don't need to write that down, but it's a particular bacterium that we do find in the human body, particularly in the gut. Back here at the beginning, we're only at about 20x or 40x in terms of our lens, which is why it's super hard to see any of these cells. If you look really carefully, you can see something. You can see little dots, and every once in a while you'll see one of them moving, but super hard to see at 20x or 40x. But this is now at 100x. We're under the 100x lens, which, if you remember, means we're at 1,000 total magnification. And now you definitely can see something. And this something is swimming around. It's moving. And that's a feature of this bacterium. Enterobacter Erogenese is modal. It has flagella, so it can swim through its environment. Now, on this view, we're also under the 100x lens. We're at 1,000 magnification. This is a different organism, though. This one's called Rhodobacter. It's specifically Rhodobacter spheroides. You don't have to know that name. I just want to point it out because you can really see, number one, there's a ton of cells. Each one of these dark blue, black colored structures is a bacterial cell. And I think you can see that they are moving around. At least most of them are. Some of them aren't. They're swimming. They're moving through this watery material that's on the slide. And again, there are a lot of bacteria that are not modal like this, but there are also bacteria that are. And so if we suspect that we might be working with a modal organism in a diagnostic sample, it makes sense to check. And we do that first by doing one of these living cell examinations. No stain, so we can see that it's moving. Here's another one for you. This is a pathogen. We're under the light microscope. We're using the oil immersion lens, the 100x lens. So this is 1,000 times magnification. You can see all these little dark objects. Some of them are clearly swimming. Some of them are not clearly swimming. That's normal. I want you to notice though, see if your eyes can follow one of these cells. I want you to notice that they're not really swimming in a straight line. They're kind of rolling. They're kind of spinning. It's the strangest type of movement. See if you can see that. Some of them are just like spinning around. This is a very particular type of motility. This is called tumbling motility. This is very characteristic of a bad pathogen called Listeria. The bacteria is called Listeria Monocytogenes. Again, you don't need to write that name down. But Listeria is a bacterium that can infect a lot of different types of food. And it's particularly dangerous for people who are pregnant and for people who have immune problems. Their immune system just isn't working as well as it should. This sample, when you look at it under the microscope with the cells alive, if we see this very characteristic tumbling motility, and again, we think we might have Listeria, this is a characteristic that you see very particularly in this pathogen. All right. All right. Questions? Any questions about what we've looked at so far? The other thing I should mention about these samples that we make, the wet mount and what's called a hanging drop sample, we'll talk more about those in a minute. They're very quick to make. And that's nice too, because sometimes we just want to get a quick idea. Do we think we have a mobile organism, a motile organism? Well, let's do a quick examination and see. It actually takes some time to make a slide and stain it. Now, we're probably going to do that too when we're in the diagnostic laboratory, because there are other things that we want to test for. But if we can quickly do an examination for motility by looking at the cells while they're alive, that can add a lot of information into our data to help us identify some of these pathogens. All right, let's look at this next slide together. There are two ways that we examine living microbes under the microscope like this. Two ways that we prepare a sample for the microscope. One is called a wet mount, and one is called a hanging drop. Now, both of those are used without stain. So we're going to use the sample, whatever the sample is. Maybe it's lake water. Maybe it's from somebody's well. Maybe it's urine. Maybe it's fecal material. But we're going to do a quick wet mount or a hanging drop. And we're going to not only be looking at the cells, but we're also going to be thinking about whether these cells are moving, whether they're mobile. Wet mounts are pretty good for seeing motility. Hanging drops are very good for seeing motility. So we'll talk about how to make both of these types of slides. Now, a couple of things to know when you're looking at living cells like this under the microscope. You have to mix up your sample really well. If you get a sample of lake water, for example, and you've got maybe 50 or 100 mLs in a jar, over time, the cells are going to settle out of that water. So it's really important that you mix up your sample before you take some out and put it on a slide. The same is true for any patient samples. You would want to mix up, for example, a urine sample so that if there are organisms down at the bottom, you're dispersing them throughout the sample before you draw out a little bit. Now, anytime you draw out some material to look at, you got to use a sterile instrument to pull the material out. You have to use either a sterile pipette or maybe a sterile loop, or we'll talk about loops a little bit later in the course. That way, you're not accidentally adding any more microbes into the sample. You want to make sure that you're being as clean as possible and you're using sterile tools to gather your materials. Now, once you have that, a little bit of the sample material drawn up, you're going to place it on a microscope slide. If you're not particularly familiar with making microscope slides, we put our sample right in the middle of the slide, generally in the middle 1/3 of the slide. And we really do just use a drop. It's very hard when you're first learning how to make slides. It's very hard not to put too much on a slide. We always want to put more than we should. And we really only need a very small amount to see what we need to see. Now, the other thing I have on this slide here is that our microscope slide should be labeled. Remember that labeling we talked about? If I'm going to draw up a sample out of, let's say, a patient urine sample, it's very important that my slide be labeled too. Remember, we talked about the kinds of things that belong on labels in the microbiology lab. Your initials or your name, the date, the type of material it is. Is it urine? Is it blood? Is it fecal material? Is it lake water? You want to make sure that that's written down on the slide so that if you put the slide down, or maybe you're making 10 different slides, you know exactly what's on that slide when you pick it up. Label, label, label. So on this slide, just a couple of images to drive home this idea. Over on the left here, this is a little Vortex machine. If you've never handled one of these, it just helps mix up samples. So you can see someone's hand here. They're holding a test tube with a blue liquid in it. If you press the test tube down into the vortexer, it's going to basically vibrate and it will mix this liquid up nicely for you. You can use different instruments to draw out a drop of material. In the center here, these are very inexpensive little, what are called transfer pipettes. And you just want to make sure that you use a sterile one so that you're not accidentally introducing microbes into this material. Now, the other thing to notice, and this is a small detail, but it's an important one. You can see this person is drawing a sample out of a beaker. Now, maybe there's 100 mLs of fluid in here. You don't want to draw your sample from the very top or from the very bottom. After you mix it up, you want to draw your sample right from the middle. It's just a technique. And if you do it the same way every time, you'll get the best sample. Remember, you've got to mix things up first. And then the best the standard technique is to draw from the center of however much of a volume you have. If you're pulling out of a tube like this one, you would just go right into the center of that volume and pull out a couple of drops. Now, if you take a look on this slide over on the left, this person is holding a microscope slide with one hand, and they have a transfer pipette in their other hand. And you can see they're just going to place a little drop right in the middle of the slide. If you were to use what's called a micropipettor, and that's a tool that can actually measure precisely how big of a drop you want to put on your slide. Generally, it's about 20 microliters. I didn't say milliliters, I said microliters. It's a tiny volume, and it's generally smaller than what you would think you need. Now, over here on the right-hand side, they've drawn that glass slide for us right here, and they're showing us the droplet of sample on the slide. So we're looking at this slide now from the side. We're not looking down on it. We're viewing it from the side. And this picture shows us how to place a cover slip correctly. Anytime you make a wet mount, you're going to put a cover slip over your drop of material. Remember, all a cover slip is a little slip of glass. It's very, very thin. It's about 1/3 the size of a microscope slide. And you're going to put it down on top of your liquid sample, but you don't want to just drop it, because if you do, you'll get a lot of air trapped under there too. So we put one edge of the cover slip onto the glass slide, and then we very slowly lower the other end down. That's going to minimize any air getting trapped under there. So you put a drop on a slide, you put your cover slip on, you're ready to go. It's very quick, very, very quick technique. Take you a couple minutes. And again, it's a nice way to just quickly determine if you have any moving cells in your sample. There are some limitations to a wet mount though, and these are important to know. Probably the biggest limitation is that this slide is going to dry out really fast. Remember, there's a tiny little volume of liquid on that slide. So under the cover slip, that little volume is going to evaporate very quickly. Generally, you've got about 15 minutes, and that's it. So you can't make a wet mount and then come back 2 hours later to look at it. You have to make it and look at it. Now, there are a couple of little techniques that people use to try to make a wet mount slide last longer, and these are pretty helpful. You can put a little bit of petroleum jelly, a little bit of Vaseline around the edges of the cover slip before you place it over your drop. It's just going to seal out some of the air that gets in there. It's not going to make it last forever, but it might give you an hour instead of 15 minutes. So you just have to know that when you make a wet mount, you then have to look at it. You can't save it for later. It's not going to last. Now, something else to know about wet mounts, and this is important when you're first learning. When you put a wet mount onto the stage of your microscope and you start examining it, you have to remember that the light that is coming up from the bottom of that stage gets hot. And you're going to kill those cells if you sit there for 15 minutes or 20 minutes looking at those cells under that hot light. So even though your wet mount will last for 15 minutes, you can't let the light just cook those cells for 15 minutes or they're going to stop moving. It gets really hot. You and I wouldn't notice how hot that light is. But if you were a little microbial cell in a little droplet of water, even the light coming up from the base of that microscope is going to be like the sun, and it will very quickly just fry them. So again, it's another reason why when you make a wet mount, you want to examine it quickly. Now, wet mounts are very, very common. People use them all the time, every day in microbiology labs. But there is another technique that you should know about, which is this hanging drop technique. This one is actually better than the wet mount for finding mobile cells. And the reason it's better is because it forces all of the cells into a smaller area, so they're easier to see. Now, you can, as I said, you can see motility on a wet mount, and a lot of people don't like making hanging drops because they're a little harder to make. They take a few more minutes to make. But like I said, if you are really kind of thinking that there's a mobile organism in one of your diagnostic samples, or a physician has said, please check to see if there are any mobile organisms in here, a hanging drop might be your best friend. It requires a slightly different slide. We don't use an ordinary microscope slide. We use a slide called a depression slide. The other term you'll hear is a well slide, W-E-L-L. These are glass slides that have, like the name suggests, a little depression cut out of them. They have a little well or a little cavity cut into the surface of the glass to hold the drop, essentially. You're also going to need a cover slip, just like with a wet mount, and you're going to need some petroleum jelly for this. Now, the other thing that a hanging drop is useful for, that a wet mount is just not useful for, is seeing if cells arrange themselves in a particular way. Now, that might sound strange to you, but we're going to talk in here about how certain bacteria like to arrange themselves into patterns when they grow. For example, Staphylococcus, which is a bacteria you've probably heard of. Staphylococcus likes to group together in these clusters, almost like a cluster of grapes. There's another kind of bacteria called Streptococcus, and that bacteria likes to arrange itself in chains. So sometimes we can see these arrangements in the hanging drop. On a wet mount, the cover slip actually compresses the sample a little bit too much for us to see these arrangements. So another reason a hanging drop might be better. So there's a lot of information on this slide, but follow along with me and we'll see if we can look at it together. Up at the top here, we're looking at a glass slide again from the side. We're looking at it on the side view. And I think you can see that there's a little piece of the glass cut out here, a little well that's been dug into the glass. Now, in this particular drawing, they're showing us a drop of water, some kind of sample. There's a cover slip on top of it, but there's also a little petroleum jelly underneath the edges of the cover slip. So there's a little space between the cover slip and the slide. Can you see that? So when we do a hanging drop, and it's really hard to see in this particular picture, but I have more for you. When we do a hanging drop, the reason we call it a hanging drop is because this droplet of liquid is not sitting on the slide. It's hanging down from the cover slip. We're not going to put the drop into the well on the slide. We're going to put the drop on the cover slip, and then we're going to flip the cover slip over and let that drop hang. It's just hanging off of the cover slip. If you look really carefully, you can see there's a little bit of space between that droplet and the well in the slide. So it's a technique that takes a little practice, but it's not super hard. I'm going to show you how to do it in just a minute. Over here, what you're looking at on the right, this is a actual picture of one of these depression slides. You can't really see very clearly, but there is a cutout well right here. If you were to feel that area with your thumb, you would feel a depression in the slide right here. Down at the bottom here, all they're showing us is that you have to have petroleum jelly around your cover slip in order to do a hanging drop correctly. Now, the primary reason we do hanging drops is because it gathers all of the cells in the sample together for us. And if those cells are moving, we're going to be able to see them. What you're looking at on the left-hand side here is what a hanging drop looks like under the microscope. I'm under a high power here, so I'm under the 100x lens here. What you're seeing on this area, this is where the drop is. Notice it doesn't look like a drop. It's got a strange sort of zigzag S-shaped line. but the water, the droplet is over here with the cells in it. And then there's just plain cover slip and space over here. Now, under the actual microscope, it looks like this. So what you're looking at over here is droplet and space. Now, this is a picture, not a video, so you can't see that these cells are swimming around. But I have another one for you where you will be able to see that. This is a short video here. I'm going to go ahead and play it, but I first just want to orient you. This is a person who is working underneath a hood. They've got a glass test tube in their hand, and they're using that tool called a loop. You can see there's a little volume of material in here. This is just some culture material. So this is bacteria have been purposefully grown in this culture material in this tube. You'll also see that there's a bunch of slides laid out over here. There's also several cover slips laid out near the slides. They've drawn circles on these slides. Okay. I did not make this video. I make most of the videos that we use in here, but this is a video that I found somewhere else. So question, what's wrong with this picture? What is wrong with that picture?
No
They're not wearing
gloves.
gloves.
No gloves. Oh, it's so hard for me to look at that. I know. I'm silly, but oh, you would never, ever hold a culture tube like this in your hands without gloves on. Bad, bad, bad technique. Maybe they were just making this for the purposes of the video. Maybe there's not actually any bacteria in there, but the person should still have gloves on. All right, I'm going to go ahead and click start on this. What you're going to notice is that this person is placing just a little tiny droplet onto each cover slip. They're not placing it onto the slide. They're placing it onto the cover slip. This is that loop tool. It has a little loop of metal on one end. It picks up a tiny volume of liquid. Now the person is using a flame to clean off that metal loop. Don't worry about that right now. Now what they're doing is they're putting a little bit of this Vaseline material around the depression area, around the well in the slide. Now, remember, they put the drop on the cover slip, not the slide. So what they're going to do is pick up the slide and press it onto the cover slip. They're kind of doing it backwards. You pick up the slide upside down, you press it onto the cover slip like that, and then you flip it back over. Now the cover slip is on top. And hopefully that droplet is hanging down into the well of that slide. Now, the person is now going to come over and work on the microscope. And as always, we first use the lowest power objective lens to find our hanging drop. And then once it's in focus, because this is a parfocal microscope, they can just go up to these higher magnifications and look for the motility. And that's what we're going to see next. So they're getting the lens in and they're looking again for something that looks like this with a partial drop and then an empty place on the slide. And this is what it looks like under low power. This is the droplet on the left. This is the empty area. Now, at low power, you're not going to see any cells. You've got to go up at least to 40x. And even better, you want to go to 100x lens. Again, the drop will be on that side, the space on this side. Now we're at 40x here. Give it just a minute. Can you see a little bit of movement in there? Just a little bit. Hard to see, I think. Out of focus, really. Now give it just a sec here and I'll pull it back. Take a look again. The cells look black. See them? There's a cell right there swimming. Hard to see, I know, hard to see. but at least you can see that there's going to be fluid on one side and then space on the other, basically air under the cover slip on the other side. And again, when they're done well, hanging drops are super useful because they draw all of the cells over to the edge of that drop. And if there's any movement, we're going to see it. All right, questions? Any questions about what we've said so far? So you put the drop on the cover slip. I don't have any organisms today, so I don't have any gloves on. Cover slips are tiny. Let me see if I can do this. They're very, very small pieces of glass. I'm going to put a droplet on the cover slip. I'm going to put this, and then I'm going to put some Vaseline on my slide. I'm going to press that Vaseline down on top and then flip the whole thing over. And with any luck, I have a droplet hanging down from that cover slip. It sounds easier than it is. When I make hanging drops, I'm successful about 2/3 of the time. I have a problem with making my drops too big. And if your drop is too big, it's not going to hang. It's just going to come down and hit the slide. It's not going to work. I personally, though, prefer a wet mount. I'd rather search for cells in a wet mount. In my hands, I'm just better at making wet mounts than I am doing hanging drops. But I do want you to know about hanging drop technique.
I would just
All
see
right.
myself getting the Vaseline every time.
Say again.
I would just see myself having to constantly hit the Vaseline every time.
Oh, it's bad. And you know, there's different ways to put the Vaseline on. I was always taught to use a wooden toothpick, but people use different things to put the Vaseline on, and it's so easy to put too much Vaseline on, and then you're just smeared with Vaseline everywhere. So it's not, it does take a little bit of practice to make them correctly. All right. I want to switch gears here now and talk a little bit about the difference between true motility, which is when a cell is actually swimming because it has an appendage called a flagella, and something that we see under the microscope called Brownian movement. Now, Brownian movement got its name because of the researcher, the scientist who first described it. What Brownian movement is the movement of cells and other particles under the microscope, not because they're moving by themselves, but because either the liquid around them is moving or they're being struck by other molecules, which is causing them to move. So either there's a little current in the liquid around them, and that's pulling them along, or there are tiny microscopic particles in there that are bumping into those cells and making them appear to move. Now, Brownian movement can be hard to distinguish from actual motility, but again, like everything else that has to do with microscopes, the more you practice, the better you get at telling it apart. Brownian movement is most problematic, most evident under the high power lens. So when you're up at 100x, when you're using your oil immersion lens, you got to remember that Brownian movement exists, and you got to really be careful that you don't mistake it for true motility. Now I want you to take a look here. This is, we're looking under the microscope at a sample. There's a material in here that is both cells and also some debris. Now, by debris, I mean the kind of things that show up in samples that are just not microbial cells, little bits of carbohydrate or little bits of protein, or maybe some crystals, anything that can show up in a sample that is not cellular. So this big clump of material over here, that's what I would call debris. What I want you to notice, and I'll play this a couple times, is that you can see some things moving across the field. Look, there's something right there. There's something right there. Can you see that? There's something there. What you're looking at here is this Brownian movement. These are not things that are swimming. These are not things that are swimming. Now you might say, well, how the heck do you know? How do you know that that's not motility? Again, it takes practice. But when you look at Brownian motion, this particular type of Brownian movement is because there is a current moving across this slide. There's a lot of liquid on this slide, and the liquid is moving. There's a little current of liquid here, and it's carrying this material along with it. The way I know that is because everything is moving in exactly the same direction at exactly the same speed. Okay. Now I can also tell things like, I can tell what looks like a cell and what doesn't. We haven't talked about that yet, so don't worry about that. But anything that you see moving in this video clip, it's all moving in the same direction, left to right, and it's moving at the same speed. That's not motility. When things are truly modal, they're going to be swimming randomly. Every cell is going to be swimming in its own path. So when you see this, very clear movement where everything is moving the same, you should suspect Brownian movement. Now, over here on this slide, right here, I have 1, 2, maybe 3 or 4 yeast cells. These are yeast cells. And once the focus stops for a second, you'll see that they're moving a little bit. So are these down here. They're just vibrating a little bit. They're not traveling anywhere. Can you see that? They're just kind of bouncing up and down. There's a little something up here too that you might think is, oh, maybe that's bacteria. It's just sort of vibrating. It's just sort of bumping up and down. This is just Brownian motion or Brownian movement. There's something in here that is just bumping in to these cells and causing them to vibrate like this. They're not traveling anywhere. They're not moving any great distance. They're just vibrating. You can see it up here too in this corner towards the end of the video clip. There's something that appears up here, a cell or some debris. I'll show you when you can see it better. And it's just sort of vibrating in place. It's not going anywhere. This thing right here, it's another yeast cell. That's very typical for Brownian movement. Here's another one. I just want you to look at what's on the slide, like over here and over here. These are yeast cells. And just notice that there's 1, 2, maybe 3 cells here, and they're all going exactly the same direction at exactly the same speed. That's not motility. They're being carried along because the liquid on the slide is moving. Let's look at that one again. I got a bunch of cells right here. There's 1, 2, 3, maybe 4 cells clumped up together here. And they're going to travel across our field of vision. But they're just sort of slowly going in this direction. They're all going exactly the same speed, exactly the same direction. They're not moving under their own power here. They're being carried along. Now compare that to what we looked at a few minutes ago. Compare that to this organism. Look at how these cells are swimming. They're not all going in the same direction. They're going in different directions. Some of them are going really fast. Some of them are going slower. Some of them are turning in circles. Some of them aren't swimming at all. But there's a real difference to seeing a cell that is moving under its own power, on this slide too, versus cells that are being carried along. See how these cells are all like living their own lives? They're swimming in their own path. They're not following each other. They're not all going in the same direction. They're not all going at the same speed. That's the difference. That's the difference between a cell that is modal, that is moving under its own power, and Brownian movement. Anybody have any questions about that? When you're learning microscopy, if you go into any field where you're working under a microscope for periods of time, it doesn't even matter what you're looking at under the microscope, whether it's microbes, or maybe you're in a pathology lab, or maybe you're in a research lab. If you're looking under a microscope at samples as your job, it's very hard at first to distinguish between true motility and Brownian movement, but you get good at it after a while. It's just like anything else. It just takes a little practice. All right. So the last thing I want to do with you today before I let you go, I want to walk through a wet mount with you. Now, this is a sample that I made. You can see in the image, I'm holding just a little plastic test tube, and it has this reddish colored liquid in it. I used a strawberry, just a plain old strawberry, and I mashed it up in a little bit of water in this test tube. So I didn't add any microbes to this tube. All I did was I took this naturally growing fruit, this strawberry that I bought at the grocery store, and I mashed it up. Strawberries and other living organisms, just like humans, have a microbiome. So they're covered in microbes too. Now, I don't mean to gross you out, but the good news is that most of the microbes that grow on a strawberry, for example, are perfectly harmless to us. In fact, the things that grow on fruits and vegetables are what allow us to ferment those fruits and vegetables. So it can be very helpful when we're using fruits and vegetables to make other food products. So I just mashed up this strawberry in a little bit of water. And now I'm opening up the test tube to draw some out. Now, I mentioned a minute ago that you want to use a well-mixed sample, that you want to use a sterile tool to pull out a drop, and you want to draw from the center of your sample, not from the top or the bottom. There's something else in this picture I want to point out to you, and you'll hear me talk about this again and again in here. Notice that I've done two things that might look unusual. I took the cap off and I kept it in my hand. I didn't put it down on the desktop over here. The reason I didn't is because I don't want this to get contaminated. I want to keep this sample as clean as possible. And if I took this cap and I set it down on the counter, I might pick up a microbe or two off of the counter. So anytime you uncap a tube in a microbiology lab like this, you want to keep it in your hand. You can just tuck it between your fingers somewhere where it's comfortable. Now, the other thing I'm doing with this tube that's important is I'm tilting it. Notice that I'm not holding it straight up and down. Remember, one of the things we said last time was microbes don't jump up, they fall down. They fall down in air currents. If we keep our sample tubes open for as short a period of time as possible, and we keep them at an angle, we will help prevent things from falling down into those tubes from the air. You're going to hear me mention that a few times in this course. So I have my labeled microscope slide here. You'll see I got my initials and a date, and I've written down that there's yeast on here because I know what is growing on my strawberry already. You'll notice that I have a drop of that strawberry liquid right in the center part of the slide. Now I'm going to place my cover slip. It's hard to see, but I'm holding that little piece of glass, that cover slip. I put the edge down onto the slide, and the drop gets sort of sucked over to the edge. Now I can very slowly drop that cover slip down, and I'll get very little air under there, which is good. Now I'm ready to examine my slide. You'll notice that I've taken my gloves off. Remember what we said? We said we don't ever want to handle anything with our gloves on that is not routinely disinfected. And one of the things we handle most often that we don't disinfect is our microscope. So once you have your slide made, once you have your sample made, gloves come off. If I'm going to touch the microscope, I want my gloves off because I don't want to accidentally contaminate my microscope. Any questions about how I made that slide? I have to put a tool in there to pull up some fluid, but I need to make sure that that tool is sterile so that I don't accidentally put anything that I don't want into my tube. I'm going to keep the cap in my fingers, in my hand, so it doesn't get contaminated. And I'm going to tilt my container so microbes don't fall into it from the air. Now, what you're looking at on this slide, this circle here is what it looks like underneath the microscope. You couldn't see on the slide a moment ago, but it's often very helpful when you're first learning how to focus a microscope. Instead of just putting the droplet in the middle of the slide, to take a Sharpie pen or a wax pen and draw a circle. Draw a circle on the slide and put your drop in that circle. Underneath the microscope, the line that you drew is going to look like this. It's going to be big and thick and black, and it's going to be very easy to find. If you can find the circle that you drew, you can find your droplet. So it's a nice little trick when you're first learning how to focus a microscope. Now we're looking under the low power lens at my strawberry liquid. So remember, on some microscopes, that's going to be a 4x lens. On some, it's going to be a 5x lens. It's just a low power lens. This is not enough magnification for me to see any microbes. But I can see other things on the slide, like debris. And there's a lot of just brown looking material on here. There's also this strange looking blue thing right here. I think this is a fiber, a clothing fiber. I'm not 100% sure though. So when we first focus, we focus under low power. And then we can move up to higher power lenses. And because our microscope is parfocal, we won't need to do much more but fine focus a little bit. And this is what we see. So what you're looking at here, and we haven't talked about different types of cells yet. That's coming this week, but these are yeast cells. So what was growing on my strawberry as part of its microbiome is a type of fungus. Now, I don't know what species of fungus it is, but I know it's not a harmful fungus. It's just a natural organism that naturally grows on the skin, if you will, of a strawberry. And underneath the microscope, I can see some interesting structural features. These branches right here that you see, that's part of the fungus. That's called hyphae. You don't have to write that down. We're going to talk more about that later on. But these branches are alive. This is part of this fungus. And what it's doing is it's creating these sort of egg-shaped cells called yeast. And they're going to bud off of these branches and go off and grow their own new branches. On this slide, we're also looking at the same strawberry sample. We're just under a different field. All I've done is I've moved the slide over a little bit underneath the lens. So I'm looking at a different area in the droplet of fluid. You can see a branch here that has 1, 2, 3, 4, 5, 6, 7, 8 different yeast cells on it. Some of these species of yeast will make several yeast cells before those yeast get released into the fluid. Very, very normal. And here's another area of the slide. I took this picture to just to show that depending on the magnification that you're at, you're going to get a very different view. You can see the chains of yeast cells here. You can see the branches that we call hyphae here. I haven't added any stain. I haven't done anything to kill the cells here. These are living cells underneath the microscope. Now take a look at this slide, because this is just another field on the same slide. You don't see any of the hyphae here. You don't see any of those egg-shaped sort of round yeast cells, but you do see these strange little things here. Now I put this slide in here very specifically to remind us that we always have to be thinking about what magnification we're at, what lens we're using, and what kind of organism we're likely seeing. Remember, we said in the homework, the reason we knew we weren't looking at a virus is because you can't see viruses under a light microscope. Well, in this image, if you were to just look at this and you didn't know anything else, you might look at this squiggly line here and you might say, gosh, I wonder if that's one of those spiral-shaped bacteria. I wonder if that's a spirochete. I wonder if those are bacterial cells that I'm looking at, shaped like a spiral. Now, if you knew nothing else about this sample, you'd be forgiven for thinking that. And there are bacteria that look like this underneath the 100x lens. But this happens to be underneath the 4x lens. And what you're looking at here are just little pieces of the hyphae that have broken off. So this is essentially little bits and pieces of that fungal branch that we looked at that have just broken away from the rest of the colony of fungus. So if I know I'm looking under 4x, there's no way these are bacteria. Remember, I can't see bacteria until at least I'm using the 40x lens, what we call the high dry lens. I can see the bacteria even better under the 100x lens or the oil immersion lens, but I'll never see bacteria under a low power lens. On this slide, I'm still looking at the strawberry liquid. I'm still under 4x lens here. All I did was I put a drop of stain on top of my drop of strawberry liquid. Now that's not the same as making a stained preparation. I didn't attach the cells to the slide with heat, and I didn't apply the stain in any particular way. I just added it. I took a drop of stain and I added it to the drop of strawberry liquid. This is a little bit of a technique that we use sometimes when we're making a wet mount. If we're not looking, if we've decided that we know whether they're mobile or not, we can add a drop of stain to our wet mount just to help us see things a little more clearly. Remember, the stain is going to kill anything on this slide that's alive. But I just want to show you, and I know we're running out of time, I just want to show you this picture. I'm under the 10x lens here. So I'm not super high magnification. This is the same branched fungal organism. You can see the chains of yeast cells, but see how much better the detail is. You can add a drop of stain to a wet mount if you're no longer interested in motility, because the stain is going to kill the cells, but at least it'll give you some more detail and help you see some of that detail a little more clearly. All right, I know we're out of time. I just wanted to quickly remind you that when we get together on Wednesday, we're going to talk about simple staining, basic staining techniques. And we'll look at different bacteria that have been stained, so you can see the different cell shapes that bacteria take. But I don't want to keep you any longer. Anybody have any questions before I let you go? I will post the recording of this lab meeting up on our playlist as soon as I download it. It does take a few minutes for me to do that, but I'll post it up to YouTube as soon as I have it, just in case you need to come back and look at something again. But that's all I have for you right now. I will see you again on Wednesday. Thanks for coming. Have a good night. We'll see you next time.